Thank you, Krista. Thank you for this invitation, and basically my lecture will be describing to you how we should be evaluating tests, how we should be demonstrating and providing the clinical evidence for the effectiveness of tests. The framework I will be presenting will be more than what the IVDR requests at the moment, but I would like to demonstrate through this framework that it's not just the manufacturer's responsibility to evaluate the tests, but it is also our responsibility in the laboratory. So, okay, let's start with a scenario. Imagine that you don't have to imagine because you are a specialist in a laboratory of a, say, metropolitan hospital. And one day a company rep from the R&D department comes to you and he just says, well, we have developed a panel of protein biomarkers, let's call them New Wonder, that may help predict coronary artery disease. So they believe that this is a much better test and an earlier marker than your current test panel, which contains all those well-known biomarkers which we all use in the laboratory, which I would just designate as old or. Okay? So we want to demonstrate to you this is far better than your old or for the risk prediction of coronary artery disease. Okay, so you go to your clinician, because you are a good laboratory professional who actually can start with the clinician. And then the clinician says, what? From a droplet of blood? You're kidding. No, I think we should be looking into it, but I don't know. That's what they claim. Okay? So you are a bit doubtful, you are a bit skeptical, which you should be before you implement a test. Especially because you are also reading the literature, of course, as a skilled laboratory professional. And in the literature of proteomics research, you already know that a lot of tests don't actually pass the biomarker translation pipeline. A lot of them really fail, and only very few, for example, according to this assessment in clinical chemistry, only 4 of 44 proteomics biomarkers were, only 4 out of the 44 proteomics biomarkers made progress to clinical application. So you should be doubtful a little bit that is this new wonder truly something that will solve your new medical need. So Krista has already introduced to you the evidence regulations, the IVD regulations, and I'm just highlighting the evidence collection part of it. And I will be only focusing in the rest of my talk on the scientific validity, analytical performance, and clinical performance of new biomarkers and how you should be investigating those. So how should you really evaluate the performance of this new wonder before you implement it into your laboratory? Now, just referring to the IVDR Article 56, a performance evaluation shall follow a defined and methodologically sound procedure for the demonstration of scientific validity, analytical performance, and clinical performance. Now, what does scientific validity mean? It means the association of an analyte with a clinical condition or a physiological state. You really have to prove it somehow, and you really have to look into it. Where does this information come from? Now, this was an R&D guy, and of course, they had that sort of information. They will bring to you this proof of concept study, whereby they have demonstrated in experimental conditions that there is a certain protein signature, I'm not going into the details of it, of several biomarkers forming a panel which will help you to identify whether there is a high-risk plaque or a low-risk plaque, or whether there is a presence of coronary plaque or there is an absence of coronary plaque, depending on which protein is overexpressed and which protein is just underregulated. So this is the new wonder they come up with, but they only had a proof of concept study to justify its use at this stage. So now, we in the test evaluation working group, we are following a so-called consequentialist approach, meaning that we are not just interested in, that there is something new and fantastic, but what is the consequence of that potential new biomarker to clinical care and to the diagnosis of patients. So therefore, we have developed this framework for evaluating new biomarkers in the working group quite a few years ago, almost 5 years ago now. And I'm going to demonstrate to you what this framework is about. And this is nothing particularly new. You know all the elements of a test evaluation framework. You know that there is an analytical performance that needs to be demonstrated. There is a clinical performance of an assay that we have to prove. We will be looking into the clinical effectiveness of the test, the cost effectiveness of the test, and the broader impact of the test on all sorts of aspects of healthcare. But what is commonly overlooked is that all these elements, let it be the analytical performance by itself, are driven by a clinical pathway of how the test is being used in clinical practice. And this is the area where we often don't go into that sort of depth and think about how the test is used in clinical practice. So the central role of the clinical pathway is something that I would like to elaborate on a bit. And before doing so, because again you are very aware of the research waste and that new potential biomarkers really cannot always come to fruition and implementation because there is a leakage all over the pipeline during the validation phase, the translation phase, the evaluation phase. So the pipeline is prone to failures, and maybe this new wonder is not going to be a wonder at all. But you really have to do, before anything you do with further developing that marker, further evaluating that marker, you should be considering the clinical needs for that marker. And what are the unmet needs for this new test? So your first question is there an unmet clinical need, and is there actually an intervention? So if I diagnose something with this new biomarker, can we actually treat the patient? Because just identifying, maybe without any intervention is not going to improve outcomes. The unmet clinical need is basically any missing or inadequately performing component of a clinical pathway. And in this particular case, for new wonder, for this coronary artery disease biomarker, we know that the traditional risk markers have modest predictive value for coronary artery disease. We also know that lipid-lowering therapy actually has not provided the expected benefits of prevention of myocardial infarction. And there is also, therefore, a need for some better predictive markers. So there is certainly an unmet need for better predictive markers. Now, in lack of time, I am not going to go through the unmet clinical need checklist that our working group has developed, but I would like to draw your attention to the publications in which it's described. And just very briefly, it consists of 4 steps. First, you really have to identify what is the unmet clinical need, verify it, and then validate the intended use of the biomarker and assess whether it's feasible. All these questions are more elaborated in this paper in CCA. By the way, all the slides will be on the website, so you don't have to worry too much about taking pictures. You can download them. So, and you can also use an interactive checklist that we have already put on the EFLM website, where you can use this unmet clinical need checklist. It's actually highly advisable to use it with any new biomarker industry is developing, because if you fail to ask the questions on this checklist, or the majority of the questions on this checklist, maybe you should just give up early in the development process and not waste time of developing a biomarker which is unlikely to be useful in practice. Now, the IVDR Article 56, I just repeat some of the key recommendations here. They are really demanding clinical evidence, and the clinical evidence should be such as to scientifically demonstrate by reference to the state of the art, it's important, so what you have now, in medicine, that the intended clinical benefits will be achieved and the device will be safe. Now, the clinical evidence derived from such performance evaluation should assure that the device is safe and the performance requirements are fulfilled under normal conditions of use. In other words, in routine use. Now, how do you define these words of the IVDR, reference to the state of the art and normal conditions of use? The best way of defining these terms is to refer to the clinical pathway. And that's why I was saying why the clinical pathway is so instrumental in even explaining clinical evidence for the clinical performance of tests. So you have to do a clinical pathway mapping. You have to think about it. What is the purpose and the role of the test, of this new test, in your existing or future clinical pathway? Now, the test role and purpose can be multiple, The test purpose usually is for diagnosis, monitoring, prognosis, etc. But the test role within a clinical pathway can be defined quite well by looking at all these different forms of pathways. For example, a new test can replace an existing test. A new test can be before the existing test, so that's a triage test. Or the new test can be added on to an existing test, and that's what we call an add-on role for the test. So you really have to think about where is this new wonder come into play in your existing prediction of coronary artery disease. So just the key messages from this particular aspect of the test evaluation pathway. Before a new test is fully evaluated, you have to define, is there an unmet clinical need? What is the intended purpose of the test? What is its role in a clinical pathway? On what patient population you want to use it? In what healthcare setting is it going to be used? And the condition that is intended to be managed with this test, as well as the procedures for evaluating those and the potential final outcomes of the testing. So what do you want to achieve with the testing in the end? So all the above questions are best mapped out if you actually draw a clinical pathway. And that one you can do with your clinicians. That's a perfect opportunity for clinical consultation. Okay, the next step is, and now I refer to the IVDR Article 2, to do an analytical performance study. And this is what we are pretty good at. What is analytical performance? We all know the ability of the device to correctly detect or measure a particular analyte. What do we do? We are pretty good at that. We consider the pre-analytical aspects, we consider analytical sensitivity, specificity, limit of detection, quantitation, blend, measurement range, linearity, cutoffs, metrological traceability, imprecision, trueness, interferences, cross-reactions. All this stuff is already in a kit insert by manufacturers because they already had to comply with this under the previous IVD regulation. So what I just would like to highlight, because we know this, we are pretty good at this, manufacturers are really good at this, but the pre-analytical bit is a little bit tricky, often forgotten. So this is just in relation to our new wonder proteomic panel. Actually, they found out, while validating this marker, that there is a significant pre-analytical component here. In the discovery study, 25 of the 653 plasma proteins they identified, they changed the concentration after heparin was given to patients. Now, bad news, because you are actually measuring an artifact, and you are displaying it as you wonder, and you are actually evaluating it thoroughly. You do all the analytical performance and clinical performance studies on something that is basically just an error, a pre-analytical error. So we have to be very careful, and now research organizations are pretty careful too. They started developing standard operating procedures for pre-analytical aspects of proteomics research, and you can find SOPs on the HUPO website to actually define how specimens should be collected and processed. There are also new standards for quality assessment of mass spec studies, and Krista was one of the key authors, for example, of the article whereby the quality requirements for quantitative proteomics have been laid down, and the new CLSI standard is underway, for example, for clinical proteomics maybe next year. So I'm just telling you that these are the things that need to be followed in addition to the IVDR, because these are the documents which tell you how to do it, what to do and how to do it. I'm just using this proteomics example. There are many other types of omics and general chemistry markers that should go through the same process. So now the key message for the analytical performance bit is that the analytical performance specifications for your test should reflect clinical needs. It can be based on 3 different models, which we will be discussing tomorrow in a session about analytical performance specifications. So the 3 models come from a Milan consensus, and they should be based on preferably on outcomes or biological variation or the state of the art. The analytical performance should be set at a level that achieves net health benefit for the patient at a reasonable cost. It should be tailored to the purpose and the role of the test that I described to you before, and should be commensurate with the impact of the laboratory test on subsequent medical decisions and actions. Now, it's very important to remember that we can strive for analytical perfection, but high-quality analytical performance doesn't necessarily guarantee high-quality clinical action, or that patients are compliant, or that the chosen treatment is going to be effective. So just having a perfect test doesn't mean that the patient is going to benefit from it. So we are far still from the clinical performance and clinical effectiveness aspect of test evaluation. The opposite is also true. Poor analytical performance of the test that plays an extremely small part in the clinical pathway may not necessarily impact patient well-being. So we really have to make sure that the analytical performance of the test is commensurate with the actual use of the test in a clinical pathway. I keep talking about clinical pathway. I hope that the message will go through that. It really has a central role. Now, moving to clinical performance in this test evaluation cycle, the IVDR article again defines what that means. It is the ability of a device to yield results that are correlated with a particular clinical condition, or a physiological or pathological process or state in accordance with the target population and intended user. How do we assess that? For example, you ask these questions. How well does this actually work in practice? In what subset of patients it is working? Is it really better than old bore? How alternative tests would compare? And the way to do this, and Patrick will explain more the study design elements, for example, is a comparative diagnostic accuracy study, whereby you compare new wonder to old bore, which is basically, the state of the art at the moment, and you do that in a consecutive patient series suspected of having the target condition, for example, outpatients with stable angina, and you subject them to new wonder as the new test, and you compare it to old bore, and then you compare the performance of both tests to a reference standard, which in this case would be coronary CT angiography, to prove whether there is a plaque or not. And then you have a blinded cross-classification for coronary artery disease, and that would be the way how you perform a diagnostic accuracy study to provide evidence, strong clinical evidence, on the clinical performance of this assay. Now, just from this article, for this proteomic biomarker, you can see that the clinical model is the existing test on the left, and the biomarker model is this multi-protein panel, and you see the AUC curves. Well, of course, it is improving a little bit with this multiple biomarker, but it is far from being perfect in ruling out coronary artery disease. For a rule-out capability, you see again old bore versus new wonder. Again, improvement, but not perfect. So these are the kind of clinical evidence data that the companies will have to provide to the regulator when they put together their portfolio about clinical performance. So the test evaluation steps that are not required by the IVDR will follow. And now anybody who is from companies, now you can go asleep, because now I'm talking about the aspect of test evaluation that is more of a responsibility at the moment of the laboratory and the clinicians. So, next step, and I will go through quickly. So that's clinical effectiveness is the next step, and clinical effectiveness refers to the ability of a test to improve outcomes relevant to the individual patient or patient population. Usually we talk about health outcomes, quality of life, morbidity, mortality. So now the questions you are asking: how does the test result affect patients' health or quality of life? What are the benefits and harms and the risks of testing? So these are the issues that you need to address, again, with studies, preferably outcome studies. Now, if we go back to this new wonder again, when these guys presented the clinical performance of the assay, as you could see with the AUC curves that I showed to you, then they actually realized themselves that there is a limitation in this study. They need more external validation in a larger cohort of patients because not all high-risk plaques actually cause harmful events, and they just want to make sure that they do a larger cohort study because the prediction model may be less suitable for subjects who have low risk coronary artery disease because their study, of course, was done on high-risk patients. This is typical for clinical performance studies that we are choosing the more sick and healthy, more or less, or the less sick, and compare them. And of course, always the effect, the size of effect of efficacy is always better in such circumstances, and it becomes much less when you move into the routine cohort and routine practice. So a diagnostic RCT would be really the best way to prove that this new wonder actually improves patient outcomes. And the question you are asking, when you ask a clinical effectiveness question for a biomarker, the question is, do patients who undergo the new test fare better in terms of health outcomes than those who have the old test? And the best way of doing it by this design, again, Patrick will give you a far better description of how to do these studies. Now, the key messages for clinical effectiveness is that the link, of course, which I didn't mention, but you all know that a test by itself is not going to cure anybody. It just helps diagnosing or predicting diseases, and then the subsequent clinical action is the one that will be curing the patient. So the link, therefore, between testing and health outcome is indirect. And again, it's dictated by the clinical pathway. What will follow after you have done the test? Improved diagnostic prognostic accuracy of the test, which I demonstrated under the clinical performance chapter, or the effects of tests on medical decisions are not necessarily indicative of patient benefit. So direct evidence of clinical effectiveness would be ideal, but under certain circumstances you can't just do an RCT for every single biomarker, even though that is the desirable study design. Now, moving quickly to cost effectiveness, of course, this is something that we are all very much involved with because we have to improve the cost effectiveness of our services. So it is basically an assessment of changes of costs in relation to changes of outcomes. Problem in laboratories is that nobody bothers about outcomes. Everybody is just pushing you to reduce the costs. But actually, cost effectiveness means that you are delivering some benefit at a reasonable cost. And of course, the benefits usually are realized out of the laboratory in the clinical boards or clinics. So the questions you are asking there, what does actually this new one, the cost, can we in the laboratory afford it? Would we save money? Does this require investment into new mass specs, for example, for this biomarker? Is it really worth it? How will this impact on our workflow and budget? And overall, is it good value for money if we do this test? And then finally, the impact of testing. We are talking about consequences of testing beyond clinical effectiveness and cost effectiveness. There are organizational impacts, there are social impacts, psychological impacts of the patient, ethical and legal consequences. So these are something that still in the laboratory when we implement the test should be thinking about. So in conclusion, after initial discovery and proof of scientific validity of potential biomarkers, we really have to consider the intended use and the consequences or impact on outcomes of that test in clinical practice. So it's not just enough to look into clinical performance, although we appreciate the effort IVD industry is already proving to at least cover a good half of the test evaluation cycle. Now, no new biomarker should be subjected to tedious evaluation and released to the market if it's unlikely that it will result in improved clinical actions and measurable outcomes. And to assess whether it is going to happen or not, you really should go back to the clinical pathway and you should be thinking about the consequences of that new test before you even move into further development and tedious evaluation. Test evaluation should be focusing on the impact of variations in analytical performance on clinical performance, because the two are also related, and this is what's regulated by the IVDR, and thereby, of course, on all of these impact on clinical effectiveness through this testing, management, outcomes, pathway. So I think I use the pathway far too often now, and I hope that you will go home with that thinking that, yes, the clinical pathway is the most important thing. But as you can see, all these wheels that are closely interlinked, they are still, it's more or less a very creaky cart. Nevertheless, we are happy to be supported by researchers and industry who are trying to push this forward, and with the help of epidemiologists and methodologists who tell us how to do better studies to prove the effectiveness of tests. And of course, we, the clinicians and the laboratory professionals, and now the regulators, we are trying to pull this cart so that hopefully, eventually, the right wheels will be put on and we will progress to better tests and better outcomes for our patients. Finally, I would like to thank the old committee. It's not the current committee because we are changing, but the old committee members who all supported the development of all these concepts that I presented to you today. So thank you very much.